Hello friends, welcome to Sam Biology class. Today we shall compare between SDS page and native page. Before that, I request all of my friends, please subscribe my YouTube channel and also watch, like and share my videos with your friends. In previous video, we discussed about SDS polyacrylamide gel electrophoresis SDS phase in greater details. In this technique, the protein sample is first boiled for 5 minutes in a sample buffer containing beta-mercaptoethanol, beta-mercapto beta-mercaptoethanol and sodium dodecyl sulfate. Sodium dodecyl sulfate SDS. Beta-mercaptoethanol is a reducing agent. Reducing agent. It reduces disulfide bridges that may be present within or between polypeptide chains. Disulfide bridges stabilize tertiary and quaternary structure of proteins. In case of oligomeric proteins, breakdown of interchain disulfide bonds result in the separation of individual subunits. Sodium dodecyl sulfate is an anionic detergent. It binds strongly to proteins and denatures it. By this treatment, all the proteins present in the sample are completely denatured. As a result, their native structure and function are destroyed. So SDS phase is not suitable for separation of proteins while retaining their native structure and function. Because the protein sample is deliberately denatured prior to electrophoresis, enzymatic activity and protein-protein interaction cannot be determined on proteins separated by SDS polyacrylamide gel electrophoresis. In addition, this technique is also not suitable for the detection of protein cofactors. All these shortcomings can be overcome by native polyacrylamide gel electrophoresis, also known as native phase. Sometimes, particularly in case of enzymes, it is necessary to separate these proteins while retaining their native structure and function. To achieve this, another technique called native polyacrylamide gel electrophoresis, native phase, has been introduced. In both SDS phase and native phase, separation is carried out in a cross-linked polyacrylamide gel matrix which act as a porous mechanical support. Because of the presence of pores of specific size, polyacrylamide gels serve as a molecular sieve, thereby improving the separation of different biomolecules based on their size. The main difference between SDS phase and native phase is that in case of SDS phase, the protein sample is completely denatured, but in native phase, the original structure and the biological activity of separated proteins are retained. In case of native pairs, the protein sample is electrophorese under non-denaturing conditions. What is non-denaturing conditions? It means the sample is not boiled prior to electrophoresis, and also beta-mercaptoethanol and sodium dodecyl sulfate are absent. So the proteins retain their native state and function. In addition, native pairs is performed at low temperature around 4 degrees centigrade in order to minimize heat-induced protein denaturation. So by native polyacrylamide gel electrophoresis, we can separate enzymes while retaining their native structure and function. After separation, the proteins can be identified without staining by adding a suitable substrate. In addition to screening out an enzyme in a complex protein mixture, native pairs is also useful for the detection of fluorescent labeled proteins in a gel, and also for the determination of the native mass and oligomeric nature of different proteins. SDS pairs fractionate proteins only based on their size. This is because the protein sample is denatured prior to electrophoretic run. Whereas in case of native pairs, proteins are separated based on their size, shape and net charge density. In addition, in SDS pairs, it is not possible to recover the proteins post-separation. Whereas in case of native pairs, separated proteins can be recovered after electrophoresis. So friends, this is all about a comparison between SDS pairs and native pairs. For more updates, subscribe my YouTube channel Sam Biology Class. Goodbye and good night.